Welcome to Dr. Gordon Keller's lab in the Princess Margaret Research Tower. The lab focuses on modeling human development and diseases using stem cells, specifically studying development and diseases of the blood, vessels, and heart. We'll talk more about their research in a bit, but first, let's meet Ian.
Hello, Ian. What is it you are doing here?
Well, hello, Gregory. What we're doing is we're about to thaw our embryonic stem cells, and we have to keep them very, very cold because without this freezing of the cells, they would not be able to survive. So what we have here is millions of our cells frozen down in a very small volume. Now what we're going to do is defrost these, and then that will allow us to then grow these cells.
Sounds fantastic.
Now we've arrived at the water bath. What we're doing is we're defrosting our cells, but not fully. But we want a small amount of ice that's left, then we can put our cold media on top of them. And the reason why we have to be very careful about this part is because a sudden change in temperature can cause the cells to be shocked and to die. So what we want from that is to control for two things: temperature and the amount of our solution that goes in and how fast that goes in. Because you don't want to change the environment that these cells are experiencing all of a sudden, because that's also a stress that they cannot handle, and then they will die as well. But once we have that, and once we have plated our cells, then we can move on to growing our cells into the cell lineages that we want. And in our case, in the Keller Lab, what we specialize in is making heart cells. So any of the cells in the heart, whether they be the beating cells or the cells that go in between those beating cells in the heart, or we can make cells of the blood system, whether it be the blood cells that carry oxygen or the cells that remove dead tissue and debris known as macrophages. So what I'm doing now is I'm mixing our embryonic stem cells with a special type of supportive cells. And what this allows us to do is to grow these cells out and expand them so that we have numbers that are high enough for us to then create enough cells that we want. So let's say if we want 100 million heart cells, we need maybe 50 million stem cells that will then become the heart cells. So what we do is, after we plate these cells, we plate them onto a 6-well plate like this, and each of these cells will be filled with, each of these wells will be filled with our cells and a specialized medium that has all the goodies that they want. So this medium has a bunch of sugars, proteins, fats, and other metals and ions that they may like to keep them healthy and happy. And we feed them for about a week, and then from there we use special molecules that we have to keep very cold, because proteins, to keep their structure and shape stable, have to be kept cold. So we use two special molecules. One is called BMP4, the other activin A. And really what these special molecules are, they're found in the embryo, and they're very, very powerful when given to stem cells, in the sense that they have the power in very small quantities to tell these cells to become either cells of the heart or cells of the blood, based on the ratios of these molecules we use, along with other cues that will tell them either to go into blood or into the heart cells, or any other cells that we could imagine, such as the brain, the liver, the kidney, and so on. So now we are about to plate our stem cells, and what I've done is I've washed the cells of their freezing media, and what we can see is that we have a very tiny pellet of our cells with this white streak over here. And that white streak is our cell pellet, and that's what we will transfer into our medium, which they enjoy, and plate them into our 6-well tissue culture dish. So this liquid that I have in the tissue culture dish, this is a special liquid known as gelatin, and what it has is a bunch of these matrix proteins. So these are proteins that enable cells to sit down properly, or to attach themselves into this plastic surface. And without this coating, we would not be able to have our cells attach happily and grow well. So this step is really important to create an optimal environment for our cells to grow in. And the interesting part about this is this gelatin, or this gelatinous substance, is exactly what you'd find from cartilage and skin, and all these really tendinous aspects of the body are rich in proteins that we're currently using to plate our cells. So what I'll do now is transfer the appropriate volume into the tube that contains my cells, and I'll move the liquid so as to evenly distribute the cells within the medium. And this part is also very important because we want equal growth of our cells distributed among these wells. And this is important to maintain good consistency between our experiments and to ensure that us and my colleagues can reproduce our work. And with that, you've seen how we go from frozen cells to cells that will eventually attach onto these 6-well plates and become colonies of embryonic stem cells, which we can then take to any cell type that we wish.
And here's an example of just that. Here we have taken these embryonic stem cells and differentiated them into cardiomyocytes. These cells are the muscle cells of the heart and enable the heart to perform its function of pumping blood to and from the lungs and to the rest of the body. In contrast, here we have an aggregate of these cardiomyocytes plated against the background of non-contractile cells.
Oh, hello, Greg. What's going on here?
Oh, hello there, sir. How's it going?
It's going well. What are you up to?
Well, I'm just about to plate some blood cells into methylcellulose colony assays. So what that is we have this gel here that's called methylcellulose, and it's mixed with a bunch of nutrients and reagents for the blood cells to be healthy. And there's also some blood cells in there, and we're going to put them into this dish, and then I'll show you guys how we can look at them in the microscope. So what we do here is we use this needle, right? Because the methylcellulose is very thick, so we have to use this needle here and extract the gel out of the tube and then plate it into the dish. And again, inside here there's about 6,000 blood cell progenitors, we call them, which are just cells that will become blood cells. So there's that in the syringe, and we just place it into the dish like that. Now we don't want bubbles in there, so I'll remove a few of those, and then I'll show you exactly how we distribute the cells. Alright, so we just spread the gel out like this. And there we have it. So there's the plated cells, and I'll go show you what they look like in the microscope. Here's the methylcellulose dish that I showed you earlier that we plated, and now I'm going to put it into this imaging microscope, and we're going to look at some cells. So we'll just place it into the microscope here, and then using this imaging software. Alright, so here we've got a picture of a red blood cell colony, so we call these erythroid cells. And you could see here the red color. So all of these cells are in that gel dish that I showed you earlier. And this is one that I had plated about 10 days ago because they need to grow in the dish. And then you can also see these myeloid cells. This is a different cell type. So we would classify this as a mixed colony because there's red blood cells as well as these myeloid cells growing. So yeah, that's how you do methylcellulose, and hopefully that gives you an idea of some of what we do in the blood group.
Awesome. Thanks, Greg.
And here's an overview of what we do here in the Keller Lab. Our embryonic stem cells are also known as pluripotent stem cells. They are called pluripotent because they can become any cell in the human body. We are interested in a specific subset of cells found in the embryo, known as the mesoderm. We are interested in these cells because they can become specific cells of different organ systems, such as those from the blood, those from the blood vessels, and those from the heart. Our goal with pluripotent stem cells is to create unlimited quantities of cells from each of these organ systems. This will enable us to better understand their function and one day create cell therapies to regenerate the damage in each of these organ systems. In the heart, in the case of a heart attack, a part of the heart wall is damaged due to a lack of oxygen. We can use the cells you saw earlier, those beating cardiomyocytes, and replace these damaged cells, restoring heart function. We can apply the same concept to the cells of the blood vessels in the condition of hemophilia A, in which there is excessive bleeding due to a clotting disorder. We can create normal blood cells using pluripotent stem cells, transplant them into the liver, and restore this deficit in clotting. We can apply the same concept to the blood system, in which you can create the cells found in bone marrow transplants, known as the hematopoietic stem cell, and effectively create an off-the-shelf bone marrow transplant. Additionally, we can create other cells of the blood system, such as those found in blood transfusions, erythrocytes, and other blood cells for other different therapies for conditions of the blood system. Thank you for listening to our presentation. We hope that we've excited you about the wonders of pluripotent stem cells and the applications that these technologies will have in the future.